Okay, at Bio, this is Mr. B, and we are on our last part of Section 1 here, and we are going to talk about our last scientists today and what their contributions were to what we know about DNA. So we've kind of looked at how DNA works, the fact that it is, in fact, our genetic material, and now we're going to look at what is the overall structure of our DNA molecule. So when we zoom in on DNA, what is it actually made of, and what does it look like? So we know today that DNA is made up of nucleotides, so that's an important word. Most likely will show up on a quiz that you have because it is an important word. So know the word nucleotide here for your quiz. And it is a monomer, which mono just means one, of nucleic acids made up of a 5-carbon sugar called deoxyribose. I think I asked you what DNA stood for, and it stood for deoxyribose. That's the first word, nucleic acid. The phosphate group, and a nitrogenous base, and nitrogenous just means it's made up of nitrogen. So this is just kind of a fancy way to just say basically a little molecule, basically a monomer is all that it is that we're going to look at. And there's 4 kinds of these nucleotides that are made up of DNA. So we have 2 types, our 4 types are, make these bigger here, adenine, guanine, cytosine, and thymine. So you're going to need to know all 4 of these: adenine, guanine, cytosine, and thymine. There's no really way to memorize those, maybe come up with one of those mnemonic devices that kind of helps out some people. But you're going to need to know these, that these are the 4 little nucleotides that make up our DNA. Or these are the 4 bases that make up the nucleotides that make up our DNA. So these are our first important part of the structure of DNA. And then our backbone of our DNA chain is formed by a sugar and a phosphate groups of each nucleotide. And these nucleotides can be joined together in any order. So there's really no rhyme or reason on why things are connected the way they are. They just kind of can be random and joined together in any order. And then kind of how, so even though they're in any order, they still follow certain rules that we have to go by. So our first one is a rule by a guy named of Chargaff, is how you say that, pretty much just how just how it looks. And he said that the percentages of guanine and cytosine are almost equal in any sample of DNA. So the guanine and the cytosine that we talked about are always equal, and therefore our other 2, adenine and thymine, are almost always equal in any sample of DNA. And therefore, we know that those 2 will kind of match up with one another. So C matches up with G, and A matches up with T. And some more kind of helpful insight on the actual structure of DNA was done by a lady named Rosalind Franklin, and I'm actually kind of surprised that your book mentions her because she kind of sometimes gets forgotten. And she used X-ray diffraction to get information about the structure of DNA. So she basically shot X-rays at it, and they bounced back, and they kind of gave her the pattern of what DNA looks like. And that's just what the next part says. She aimed an X-ray beam at concentrated DNA samples and recorded the scattering patterns of the X-rays on the film. So she didn't actually get a picture of the actual DNA. She got a picture of what was bounced back from the DNA. And again, it's important to know her name, Rosalind Franklin. She was a woman, and that was kind of important at this time because, and this is the 1950s, and women in science didn't exactly mix. So she was kind of a trailblazer for women scientists. And then using clues from Franklin's pattern, James Watson and Francis Crick built a model that explained how DNA carried information and could be copied. So these 2, Watson and Crick, usually get all the glory for coming up with the fact that our DNA is a double helix. But they really kind of almost stole it from Franklin. It says they used clues, but they basically just stole it. And Watson and Crick's model of DNA was a double helix in which 2 strands were wound around each other. And so we will take a look at the picture of what this looks like. So double meaning 2, and helix is just kind of the shape that it forms. So here is our shape of our DNA here. As you can see, it wraps around each other in our double helix, and we can see that our A's, if we look at the bottom, adenine and thymine are matched up with each other, our nucleotides. And then we have our cytosine and our guanine, they are matched up with each other as well right here. So these bases are always in the same number because they're matched up right across from each other, and they basically hold our DNA together. Our A's and our T's and our C's and our G's hold our DNA together. And then Watson and Crick also discovered, kind of to end our first section here, that hydrogen bonds can form only between certain base pairs, adenine and thymine, and guanine and cytosine. Again, I just said that these two always match up together, these two always match up together. And you can see C's and G's, A's and T's, C's and G's, A's and T's. They all match up with each other. And this principle is called base pairing. So the fact that adenine matches up with thymine and cytosine matches up with guanine is called base pairing. So that's it for section 1. Let me know if you have any questions.